this week. Jonathan, quick reminder, yes, here we go, the recording button. Thank you for that. It's a great pleasure to have Patricia Cardoso with us today. So she is a biologist and she did her PhD in 2005 in estuarine ecology. She specialized in long-term studies on benthic communities in response to multiple stresses, biodiversity and ecosystem functioning. And then since 2015, she's a senior researcher at CMAR. I hope I pronounced that correctly. And there she's working on combined effects of climate change and emergent contaminants. And I think this is also the topic we're going to hear more about today. So thank you very much for joining us, and the floor is yours.
Thank you, Stephanie. Good afternoon, everyone, and thank you for being here. It's a pleasure for me to share some of my studies, my research with you. And so, as Stephanie told, my topic is about the interactions about climate change and steroid hormones. And their implications on the aquatic ecosystems functioning. So how can I, okay. So I'm a senior researcher at CMAR. For those that don't know CMAR is the Interdisciplinary Center of Marine and Environmental Research, which you can see here a picture. It's a very beautiful building in the north of Portugal, in this city called Matosinhos. And CMAR, it has three main thematic lines: the biology, aquaculture and seafood quality, the marine biotechnology, and global change and ecosystem service, where I belong to. So in these last years, I have been dedicated to the study of these combined effects of climate change and emergent compounds in these estuarine or more coastal ecosystems, including these estuarine systems, which represent around 8% of the oceanic area. They are very rich in ecosystem service, like in the provision of food and shelter for many species. They are also quite important in stabilization of sediments, in the nutrient cycling, also important areas of migration for many other species of birds. And another important characteristic of these areas, which are classified as nursery areas for many other species of invertebrates and fish and so on. So these systems are particularly vulnerable because they are quite attractive for population for living, and sometimes they are quite overpopulated, and as consequence, they are subject to many threats. So some of these threats, of course, are climate change, the overpopulation, different types of pollution. Today we are focusing on this chemical contamination, chemical pollution, and also the appearance of invasive species is one of the threats that they are exposed. So these are some of them, very general ones. When you talk about climate change, of course, the increase of temperature is something that comes to our mind, and it's expectable an increase of temperature of seawater between 1.6 degrees and 6 degrees until the end of this century. As you can see here in this picture, that is quite visible, this increase of the atmospheric temperature in the last decades. Of course, this increase in the atmospheric temperature is reflected on the ocean's temperature, of course, and and as a consequence, we are observing this phenomenon of ice melting, modifications in the ocean, in the current circulation, also the problematic of the ocean acidification, and also modification in the nutrients levels. Another topic that is less studied and it's becoming quite relevant are the extreme events. And in this case, we have done some studies, also some including the heat wave, studying the heat waves. We can say that the heat wave is considered when a maximum temperature is higher in 5 degrees Celsius to the daily average, at least during 6 consecutive days in a certain site. And what we are observing is, in fact, an increase in the heat wave frequency, duration, the length of the annual heat wave season also is increasing, and also the intensity. So in the last decades, we are observing this increase in these different parameters. So of course, we can expect some negative consequence of these heat waves, like higher mortality rates of some species that live in these habitats and cannot support this increase of temperature during so many days. Also, we are facing this displacement of some species from, for example, subtropical areas to more temperate areas. And also, this increase of temperature also favors the appearance of algal blooms, which can have strong negative effects on these ecosystems due to the negative impact that they can cause on seagrass meadows. For sure, all of you know the importance of these seagrass meadows in terms of the biodiversity, in terms of the functioning of these ecosystems. So the appearance of these algal blooms can be quite problematic. On the other hand, one of the type of contamination that I have been more focused on is on this chemical contamination by endocrine disruptor compounds, which can be which some examples are represented in this picture. So we are talking about pharmaceuticals, pesticides, plastics, and so on, which represent a group of synthetic or natural compounds with the ability to interact with the endocrine system. So when we focus on the pharmaceuticals, we know that its consumption is increasing. So a particular attention has been given to these compounds in the last decades. And the steroid hormones are a specific group of pharmaceuticals which are one of the most potent endocrine disruptors due to the negative effects that they can cause, particularly on the reproduction of these species, even at low concentrations in the range of nanograms per liter. So, of course, they can cause an environmental impact, particularly because the wastewater treatment plants are not totally efficient in the removal of these compounds. So there is a risk for the aquatic communities. In the following studies that I will present, so I have focused my work on these type of emergent compounds, which are the progestins or synthetic progestins. And what are they? They are steroid hormones with progestogenic activity. So basically, they mimic the activity of the natural progesterone. So these synthetic progestins, they are used in contraception treatments, isolated or in combination with estrogens, and can be also used in human and veterinary medicine, for example, in hormonal replacement therapies. So one of the biggest concerns about these hormones is the increase of the use of these proteins in the future, since the human population is increasing. So it is expected also to occur an increase in the use of these compounds in the near future, and of course the negative effects that they can cause on the aquatic system. Just for you to have an idea about the environmental concentrations on these compounds, we have here in the upper graph the surface water concentrations of the different proteins, and we can say that the concentration, they range from few nanograms per liter to 100 something nanograms per liter. So we are talking about few concentrations, small concentrations. When we talk about the animal farm wastewater, so we can see that concentrations are a bit higher, okay? In this schematic representation, so you can see some external factors that can interact with the reproduction, namely fish reproduction, like temperature, that can act on the hypothalamus pituitary gonad axis, as well as the synthetic progestins. So the synthetic progestins, basically, they will act at the same interconnect axis, the hypothalamus pituitary gonad axis. So these progestins, they can regulate these reproductive axis similarly to the natural progesterone, inhibiting the production of this gonadotropin releasing hormone, and of course preventing the ovulation. On the other hand, the high water temperature can also impair the gonadal development and block the spawning. So that's why it makes sense to combine these two factors and evaluate the combined effect on the namely on the reproduction of these coastal species. So what are the impacts in the aquatic life? So I will start to show you some recent data about spatial characterization of the protein levels most consumed in Portugal. So we have done this study in three main estuaries in Portugal: Ria de Aveiro, which is located more north, the Tagus estuary, and in the south, the Ria Formosa. And the studied proteins were these four: drospirenone, which is considered a fourth generation protein, so it's one of the most recent ones; desostrel and gistudine, which are classified as the third generation proteins; and the oldest one, the levonorgestrel, which is a first generation protein. So this means that, so just to explain for those that are not familiar with this topic, the first generation proteins like levonorgestrel, they were not so specific like the most recent ones. So basically they could bind to different receptors like progesterone receptors, estrogenic receptors, androgenic receptors. So they could have higher side effects than the ones like drospirenone, which is a more recent one. So is classified as the fourth generation progesterone, which is much more specific. So it binds basically on progesterone receptors. So it causes at least less side effects than the first one. So the results obtained in these three estuaries, we could see that the drospirenone and the desostrel were the most abundant ones, probably because they are the most recent ones, so they are the ones that are more prescribed. Also, what we can see in a temporal scale basically is that the drospirenone So drospirenone and zostrel were the most abundant. Drospirenone was only present in the summer, so we could just detect this one in the summer. And curiously, was more abundant also in the south, okay, in the Tagus and Ria Formosa. So what we can say here is that some values can be comparable, some concentrations of this protein can be comparable to the concentration close to the wastewater treatment plants effluents, and the other ones similar to the ones found in livestock farms. On the other hand, we can admit that the higher presence of drospirenone in the south and also in the summer can be, to a certain point, relate to some touristic pressure. Because for those, I don't know, for those that don't know Portugal, the south, which is the region called Algarve, is very famous for their beaches. And in the summer, they are overpopulated of tourists. And there is a great percentage of British tourists. And after searching a little bit about some reports on these compounds which are prescribed, we could find that drospirenone was one of the more prescribed in the UK. So we can, I think that makes sense to correlate in certain point the higher occurrence of this drospirenone in the south and in the summer associated to this phenomenon. So basically, we can say that it's important to continue to monitor these aquatic ecosystems and, of course, alert to the lack of efficiency of the wastewater treatment plants. Then I'd like to show you the results of one experiment that we did with the zebrafish, in which we have exposed them to an increase of temperature and to the levonorgestrel and evaluated effects on the reproduction and dynamics of gonad maturation. So basically, this is a picture of our experiment. This was done some years ago. The organisms were exposed to two different temperatures, 27 degrees and 30 degrees, and two different concentrations of levonorgestrel, 10 nanograms per liter, which is an environmental concentration, corresponds to an environmental concentration, and a super environmental one of 1000 nanograms per liter. So basically, they were exposed to these conditions during 21 days, and after this, they were let to reproduce. And regarding the results, they are quite curious, quite interesting, because you can see here that the organisms that the fishes that were exposed to the lower concentrations of levonorgestrel, so we could observe a significant decline in the fecundity comparatively to the control ones. And in combination with the increase of temperature, they didn't reproduce at all. So these ones, and of course the ones exposed to the higher concentration. But if we imagine those that are exposed to the environmental concentrations, when exposed to +3 degrees, they didn't reproduce at all. So there was a significant reduction of the fecundity with exposure to LNG and increase of temperature. So this protein had a higher negative effect than temperature on the fecundity of zebrafish. In this slide, I would like to highlight, you can see, so from A to D, A is a control situation. And if you go from A to D, you can see a decline in the number of late mature oocytes. So it means that there is a delay. So there is a delay in the maturation of the gonad. Okay. with the exposure to the protein, and also yes, in this case it's just with the exposure to the protein in this one in the lowest concentration, and D correspond to the highest concentration. So we can say that in terms of the reproduction, it was negatively affected by both stress factors. LNG had a stronger effect than temperature, but temperature as a single factor also caused a substantial reduction in fecundity. And in association with the LNG, we observed a synergistic effect since there was a complete lack of reproduction of the zebrafish. In terms of gonads maturation dynamics, we could observe a dose-dependent effect with the exposure to LNG. So the higher the concentration, the lower the maturation level of the gonads. And this effect seems to be exacerbated when exposed to an increase of temperature. And then we also tested these effects of combining increase of temperature and mixtures of EDCs on the condition and dynamics of gonads maturation of this gastropod, the Nucella lapillus. So quite similar, well, a little bit more complex experiment in which the adults of Nucella lapillus were exposed to temperatures, the environmental ambient temperature and the scenario of warming, and then they were exposed to a combination of endocrine disruptors, mercury, drospirenone, and of course the mixtures of both. In this case, in terms of survival, the results that we obtained were these. So at the environmental temperature, there was a higher survival, 90%. Since we increased the temperature, the survival decreased to 65 to 80%. And when exposed to the mixtures, survival also around between 74 to 79%. Observing now the maturation of the gonads, on the left you have the female gonad and on the right the male gonad. You can also observe a similar pattern. So from the control to the exposure to the mixtures, we can observe a delay in the maturation of the gonads. So here you can see these late mature oocytes, and when exposed to the mixtures of the contaminants, we observe much less mature oocytes. Okay, and the same regarding the gonad. So the exposure to these EDCs and temperature, also we could observe a synergistic effect with the reduction of the more mature stage and the appearance of less mature stage. As a summary, so these females in stage 4, so which are the more mature one, we could see 80% of them were in solvent control, but when exposed to these, for example, when exposed to drospirenone, just 40%. When exposed to mercury, was around 50%, and when they were exposed to the mixtures of drospirenone and mercury, it was reduced to 20 to 30%. So once again, we observe a similar pattern that the combination of mercury, drospirenone, and higher temperature had a synergistic effect with a delay on the gonad maturation, particularly. So this effect is more visible on females. So we can say here through this study, and according also with the literature, it seems that these proteins of last generations, so this which is the drospirenone, they have lower, that they have lower negative impact on the environment than the oldest ones, like the Lebanon shell. So and this makes sense according to what is published already, and also according to the nature of these compounds. So, as a take-home message, I just would like to say that in fact climate change and temperature increase is something that is unequivocal, and what we are observing is that the increase of temperature exacerbates the negative effect of other stress factors. In terms of the steroid hormones, namely the progestins, they have a high negative impact on the environment, but it seems that these ones that are recent ones are less aggressive to non-target species, which is good. So, of course, it is important to continue to monitor these emergent compounds, especially to raise the awareness of the authorities in terms of controlling these levels of these compounds and others which are discharged in the environment. So these are some of the more recent papers about this topic. And of course, I'd like to thank to all these funding entities and CIMAR and the University of Porto for the support to these studies. And thank you all for listening.
Thank you very much. Oh, a round of applause from everyone.
Thank you.
Yeah, thank you so much for highlighting this important topic that I think lots of us have never really thought about, and also really interesting to see that these two factors are, in concert, exacerbate the situation. So an important reminder to look at these stressors maybe together.
Yeah.
Do we have some questions? Feel free to raise your hand. I saw Julian. You want to go ahead, Julian?
Yes. Hello. Thank you for a really interesting presentation. That was great. I just wondered, when you were looking at the invertebrates, the temperatures you used were quite a lot lower than the ones for the vertebrates.
Yes, because
and you were seeing the estrogens were the new and the old estrogens had different effects, and I just wondered how that temperature difference would.
Well, basically because they are from different origins. So the invertebrate is from our coast here, so which is a temperate system. And we have used the 18 degrees, which corresponds to a summer temperature, an average summer temperature of these waters. While the zebrafish originally it is from tropical systems, so that's why we have changed it. So we have used 27 degrees as the ambient temperature, which corresponds more or less to the temperature in their origin, and so that's why these differences. So it is not a species from here.
Okay, thank you. I'm not a biologist.
Okay, good.
Thanks for that question, Jillian. Sometimes it's good to hear from the non-biologists as well. We have some questions in the chat. First, Irene, what other water parameters can perhaps exacerbate the effect of those compounds? And will the combined effect become even more complex then, when you keep adding on factors?
Other factors that can exacerbate, so it's a good question. Well, we have been testing just the temperature. We also had an experiment in which we combine temperature and also pH, so to simulate the ocean acidification. So we have so it was temperature, this was some years ago, so I'm trying to remember. It was temperature combined with this pH and also the LNG, and we tested this in a crustacean, the Gamarus locusta. But in fact, the temperature it has a higher effect than the others. But of course, can occur these synergistic effects. Others, I don't know. I didn't test, for example, effects of salinity combined with temperature or oxygen. That is also an important issue, the hypoxia and so on, for sure. But I'm not sure. I don't know exactly the answer.
Thanks. So there's still work to do, which is good. Otherwise, we
Yes.
would be unemployed, right? Next question in the chat from Salom. Interesting presentation, thanks. What are the possible routes of entry of EDCs in the biological systems?
Sorry, Stephanie, because my girl just went,
Oh.
just came here.
No worries.
Vai lá ter papá. Sorry, I was saying that, Stephanie, that today it's holiday here in my town, in Coimbra. So the kids are at home, so sometimes they can appear.
That's no problem at all.
Can you repeat? Sorry about that.
Of course. So what are the possible routes of entry of EDCs into the biological systems?
What are the possible routes
of entry? So how do they get ingested,
okay.
I guess, or
no, but
I don't know.
Basically because they are they are metabolized by our body and discharged for the water system, go directly to the wastewater treatment plants. But since they are not efficient in the removal of these and other pharmaceuticals on these molecules, what happens is that they are constantly being discharged to the aquatic systems, to the rivers, to the estuaries, and so on. So it's like this, the route.
Is that? I hope that's answering your question, Salom. Otherwise, let us know. In the meantime, Mike raised his hand.
Okay, thanks. Thanks, Patricia. That was great. It's really interesting. I shan't go into the habits of British holidaymakers and what they leave in your waters or not leave in your waters, especially in the Algarve. I would be on, as we say, very shaky ground, I think there. Although it's really interesting because as you were saying it, I was there thinking about the different compounds from different countries. But
Yes.
you mentioned your experiment with dog whelks with Nucella or Tias. Yeah, and you use mercury as well. Now, as far as I know, I'm not sure there are mercury routes into the Tagus or into the Rio Formosa, but certainly in Aveiro, in the Rio and Aveiro, there used to be there was a mercury discharge there, wasn't there?
That's right, Mike. Yeah. So for the environmental study, for the monitorization, we have chosen those 3 sites. But Aveiro, yes, it's the one that is more characterized. Rio de Aveiro is characterized by this historical contamination by mercury. In Rio Formosa, no. So in Tagus, yes, also, because it's a quite industrial area and so on. But in Rio Formosa, it was more to observe of the impact of the human population. So because in this project we wanted to study not only, so it was both the mercury and also these progestins. That's why we have chosen these areas. But you were talking about also the Nucella. I didn't understand.
Yes, no, just the experiments you used with mercury. The mercury was in that was in the zebrafish, was it?
No, in the zebrafish was just they were just exposed to temperature and Lebanon trail.
Yeah.
The Nucella, the dog whelk was exposed to mercury.
Yeah.
Yeah, because we just wanted to know, since mercury can also behave as a estrogen, can have estrogenic behavior. We wanted to test this combination, how it could combine in term with the progestins,
Yeah.
and how it could be the effects.
The mercury you used, was it inorganic mercury or was it organic mercury, like methyl mercury? Because going back to Salam's question, the methyl mercury may be taken up, the organic mercury may be taken up more easily than the inorganic mercury.
Yeah, no, it was inorganic, I think.
Yeah. Okay.
But it was not the organic form.
That's fine. Yeah, because in things like the Rio in Navarro, which has got this historical mercury, it may be the mercury is in, well, now the mercury will probably be in the sediments because I don't think it's coming out. So and if it's there, then it may be in the organic form and things. But I think this is what Salom was getting at. It's the route of uptake into organisms, whether it goes through whether it would go through their skin or whether it goes through their food or whether it goes through their gills, or they'll probably get through their gills, I guess, in most cases.
Yes.
Salom is nodding. I think.
Okay. Yeah.
Thanks for clarifying.
Thanks.
That's great. Thanks. That's good. That's good.
Excellent. Do we have any further questions? If not, then please join again in a round of applause and a thank you for this really interesting presentation from Patricia. And then I'll hand over to Jonathan to announce the next webinar.
Great. Yeah, thanks, Steffi. So we are now going to take a short break for the summer. And then it is our big ECSA 60 conference in September. So the next webinar will not be until the 3rd of October, which will be given by Victoria Mason, who was the winner of our Peter Jones Memorial Award last year. For those who aren't aware, the Peter Jones Award is for a PhD student or early career researcher that has published a paper that's made a significant contribution to knowledge. So you never know, it could be you next year. The information and deadline for submission is in the autumn. So head over to the website and take a look. And Victoria will be telling us all about the research that was part of that publication then. So I guess have a good summer. And I look forward to seeing everyone in the autumn as well.
Everyone, thanks for joining.
Thank you.
Thanks a lot. Bye.
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 Bye.